Hi class, so welcome to the second video on macromolecules. In the first video, remember, we looked at how monomers come together to form polymers. We also looked at carbohydrates and the three really important carbohydrates you need to know of starch, glycogen, and cellulose. And now we're going to finish and look at the last three macromolecules. So let's start with nucleic acids. Nucleic acids are composed of sugars, carbon, hydrogen, and oxygen, remember, from carbohydrates. They're also composed of phosphates, so phosphorus is an important element for that, and nitrogenous bases, so nitrogen is an important atom for that. And remember, way back in video 1, we talked about those 6 really important elements, chnops, and so we're always going to see these in macromolecules because these are the 6 most important elements in living systems. So the monomer of nucleic acids we call a nucleotide, and there are 4 names for these nucleotides, or 4 different nucleotides: adenine, guanine, cytosine, and thymine. And each nucleotide is composed of a phosphate group, a sugar, and then this nitrogenous base. And when we put a lot of monomers together, we call that polymer a polynucleotide. And the 2 examples of polynucleotides are DNA and RNA. And so let's look at nucleic acid structure. In particular, a nucleotide, we've got different types of nucleotides. So A and G, we consider them purines because they have 2 rings. And then the pyrimides only have 1 ring: cytosine, uracil, and thymine. And so what I'm circling here, these are the nitrogenous bases. So let's go back to this slide here. Okay, so here we have our phosphate, our sugar, and the sugar is bonded to the nitrogenous base. So this is an example of a purine because this nitrogenous base has 2 rings. But each nucleotide has these 3 components: 1, 2, and 3. And so I do need you to know that. Okay, and so we've got purines versus pyrimidines. Now DNA, when we get a lot of monomers together, we have a polynucleotide of DNA, deoxyribonucleic acid. So DNA in particular has the deoxyribose sugar. It is a double helix, so it's got 2 strands or 2 backbones, and the bases are A, T, C, and G. And that would be an example of DNA. Now RNA is a little bit different. Instead of having deoxyribose, it has the sugar ribose. It's only a single helix. And instead of thymine, it has the base uracil. And again, I hope some of this is coming back to you from your freshman biology. So RNA is simply a single strand, not a double strand. Here's another figure comparing DNA to RNA. DNA double strand, we have T, and we have the sugar deoxyribose. RNA, a single strand, we have the base U, and we have the sugar ribose. So those are 3 big differences. Function of nucleic acid. DNA, it carries genetic information from generation to generation. So your parents pass their DNA on to you. Each one of your individual cells, when it makes new cells, it passes on its DNA to the next generation of cells. RNA has lots of different functions, but probably the most important is that it's going to code for proteins. So it's going to take that DNA and turn it into proteins. It's sort of the messenger, right? It's that go-between. And so proteins then are going to determine specific characteristics in, say, a human, for example. And so what makes humans different from one another? What makes living systems different from one another? They all have DNA, they all have RNA, they all have the same 5 basic nucleotides. What makes a yeast different than, say, a plant or a human? Well, all of the variation is in the sequence and the length of nucleotides. How those A's and T's and C's and G's are actually sequenced accounts for all of the differences in life that you see. So just looking at this sequence is going to tell us the variation. Very fascinating. Again, like we saw with carbohydrates, with nucleic acids, structure fits function. So when we look at the backbone of that DNA molecule, this backbone is a sugar phosphate backbone. Very, very strong. Hard to break those bonds. But the ladder part, the rungs of the ladder, say that you would have to climb up. Those are hydrogen bonds. And remember from chemistry, hydrogen bonds are actually pretty weak bonds. So they're easily broken, which is really important because when we need to pass on our DNA, we need to copy it. And in order to copy it, we have to pull those that ladder apart. We have to break those hydrogen bonds. So structurally and functionally, it makes sense that those are hydrogen bonds and they're easy to break, easy to pull apart. Also structure. Nucleic acids have a certain directionality. So we call that being anti-parallel. So one end is running 5' to 3', and the other end is running the opposite way. And so this determines which nucleotides are added and where they're added. And this also determines the direction of transcription from DNA to RNA. So we're going to look more at that part, that structure part, later on in the year. Okay, so here's the example, 3' down here, 5' up here, and this is going the opposite direction. And this right here, the easily broken hydrogen bond, so that we can pull those strands apart, and then we can copy them for the next generation. Again, quick connection. How are we getting all of these needed nutrients and elements to make nucleic acids? Well, they first have to take them up from the environment. Just like with Big Idea 2, we need energy and building blocks in order to grow and reproduce. And so then they build the nucleotides. So plants get the nitrogen and the phosphorus from the soil. They get the sugar component from their photosynthesis products. And animals, well, we have to eat it, don't we? And we actually consume nucleotides anytime that we eat meat because we're eating something living. When we eat sugars, we get sugars to build that sugar phosphate backbone. So everything we're consuming, our bodies and our cells are using to build these macromolecules. All right, third macromolecule are lipids. Lipids are only composed of carbon and hydrogen, and they are mostly non-polar. They're hydrophobic, meaning they fear water. There are no exact monomer subunits, so we actually don't consider lipids to be a polymer. It's the only one not considered to be a polymer. And the lipids that we're going to look at in this video are fats, phospholipids, and steroids. Okay, so here is an example of a fat. All of these here, all of these points are simply carbons. And so what's implied is that you understand that coming off of each of these carbons are hydrogens. So these are simply called hydrocarbon tails. Hydro for the hydrogen, carbon for the carbon. And so these are very, very nonpolar because they're just repeating units of hydrogen and carbon. There's no charge there. And we call these fatty acid tails. And so this is the head of the fat, this is a glycerol molecule, and these are your 3 fatty acid tails. Again, very hydrophobic, they don't like water because they are non-polar. So that is a fat. A steroid is actually a lipid. A lot of people think of it as a protein, but it's not. It's a lipid. And steroids, for example, this is cholesterol. This is a fat. It's a lipid. And it is composed of 4 fused rings. So here are 4 fused ring structures. Anytime you see this sort of structure, you know you're looking at a steroid, which is a lipid. Phospholipid is probably the most important lipid we're going to look at because it makes up every single one of your cell membranes. So here's a phospholipid. It has a phosphate head, which is polar. Because it's polar, it's hydrophilic, so it loves water. But then it has these 2 fatty acid tails. And remember, fatty acids are nonpolar. They're hydrophobic. They're water-fearing. So every phospholipid has a phosphate head and 2 fatty acid tails. Remember, though, fats had those 3 fatty acid tails. So phospholipid function, it is the major component of all cell membranes. We call the cell membrane a phospholipid bilayer. Bi means 2 because it has 2 layers, 1 layer and 2 layers. And so they sort of sit on top of each other. So here are the polar phosphate heads on the outside. So this would be like your watery environment. And then inside is your non-polar fatty acid region, which makes up the inside of your cell membrane. So we're going to look more at cell membranes when we look at cells in a few weeks. But just to preview, here's our cell membrane, here's our phospholipid bilayer, another sort of look at that, and then each of these little guys is a phospholipid with a phosphate head and 2 fatty acid tails. So structure fits function again with phospholipids. The polar regions interact with other polar molecules outside, such as water, and the non-polar regions are going to differ in their saturation and fluidity. So we've got 2 types. We've got saturated versus unsaturated fatty acids. A saturated fatty acid has no double bonds. Okay, so we can see here the carbons are only singly bonded to one another. What this does is it makes them packed very tightly together, packed together like sardines. And we're going to take a look in class as to why that's not such a good thing. Now, unsaturated fatty acids have a double bond. I call it a kink. And so this causes more space to open up, because instead of being packed together all straight, you now have a kink. And so there's room there for the membrane to be more fluid. So unsaturated fatty acids are definitely better for you because they leave more room, they're more fluid. Okay, so this applies to all lipids. So saturated, no double bond. Unsaturated, yes, double bonds. So this is good to have a mix of saturated and unsaturated so that your membrane remains fluid. And we'll look at that when we look at cells. But if it's saturated, those membranes, those phospholipids are too tightly together. Okay, proteins, I'm going to go on and we're going to do that in video number 3 of macromolecules. Again, no questions, but please take time to study, study, study.